Chapter 27, Protein Metabolism. In this part, we will discuss protein translation process that takes place in ribosomes. Protein biosynthesis takes place in 5 stages. In the first stage, amino acids are activated by transfer RNA, and this results in the formation of amino acyl tRNA. In the second stage, initiation happens. In this stage, both the amino acyl tRNA and the mRNA binds to the small subunit of ribosomes. Following this, a large subunit of ribosome binds to this complex. Once this happens, stage 3 kicks in. In stage 3, elongation of the nascent polypeptide takes place. And here, covalent attachment of successive amino acid units is carried out by the ribosome and correctly positioned by the tRNA that brings in new amino acids. Elongation requires various cytosolic proteins and elongation factors. Following elongation, the fourth step or the fourth stage is termination and ribosome recycling. Completion of the polypeptide chain is signaled by termination codon. Remember, the mRNA has a start codon and a stop codon, as shown here. Termination is signaled by the stop codon. The new polypeptide that is formed is released from the ribosome, and this is aided by release factors, and the ribosome is recycled for the next step of protein synthesis. Following this, stage 5 kicks in, and stage 5 involves folding and post-translational processing. To achieve its biologically active form, the polypeptide that comes out of the ribosome must fold into a 3-dimensional conformation, and this 3-dimensional conformation must be proper and of the lowest energy. And this happens during the protein folding stage. Let us consider the composition and mass of ribosomes in prokaryotes and eukaryotes. In E. coli, the cell contains about 15,000 or more ribosomes. The bacterial ribosomes contain about 65% RNA and 35% protein. The bacterial ribosomes have 2 different subunits, a larger 50S subunit and a smaller 30S subunits. Ribosome subunits are identified by their Swedberg units, or S. Now, the Swedberg units are sedimentation coefficients. Now, the sedimentation coefficients refer to their rate of sedimentation in a centrifuge. So the S values are not additive when subunits are combined because S values are approximately proportional to the 2/3 power of molecular weight and are also slightly affected by shape. In brief, the ribosome subunits are designated with an S at the end of the number. The eukaryotic ribosome is much larger and complex as compared to prokaryotic ribosomes. And they have a diameter of about 23 nanometers in eukaryotic ribosomes. Now, as compared to bacterial ribosomes, they are only 18 nanometers. Eukaryotic ribosomes also have 2 subunits as shown here. One is a 60S subunit and the smaller one is a 40S subunit. Before discussing the 5 stages of protein biosynthesis in ribosomes, it is important to understand the structure and function of tRNAs or transfer RNAs. Shown here is a structure of a transfer RNA. One of the characteristics of tRNAs is that they are single-stranded and of about 73 to 93 nucleotides in both bacteria and eukaryotes. They have a clover leaf structure, as shown here, in 2 dimensions, and a twisted L shape in 3 dimensions. Most tRNA molecules have a guanine at the 5' end, as shown here, and all of them have a CCA at the 3' end. And this is the amino acid arm. In addition, most tRNAs have modified bases. As you can see, some bases are methylated, and some have different kinds of structure in their bases, and we'll talk about this. There are different regions in a tRNA that needs to be explained. First, the tRNA has a 3' amino acid arm, which has amino acid esterified via carboxyl group to the 2' hydroxy or 3' hydroxy of the terminal adenine of the CCA codon. tRNA has an anticodon arm, and this has 3 nucleotides that are complementary to the codon of mRNA. It has a D arm that is shown here, contains dihydrouridine, and this particular D arm contributes to folding. Finally, it has a T-Psi-C arm. It contains the unnatural base pseudouridine, has bonding between base and ribose. This is important, and this arm also helps in folding. Attaching amino acids to tRNA is an important task, and it is performed by amino-acyl tRNA synthetases. These are enzymes that esterify 20 amino acids to corresponding tRNAs. tRNAs attach amino acids via the amino acid arm. The terminal adenosine attaches an amino acid via an ester bond through the carboxyl end of this amino acid. And this can be attached to either the 3' hydroxy or the 2' hydroxy of the adenosine. The function of amino-acyl tRNA synthetases is to attach the correct amino acids to their tRNAs. Each enzyme binds a specific amino acid and the matching tRNA. Most cells contain 20 different amino-acyl tRNA synthetases, 1 for each amino acid. Some cells contain fewer than 20 tRNA synthetases, and in this case, one amino acid is converted to another after charging the tRNA. And shown here is a tRNA that is charged by an amino acid. And this ester bond is called as an amino-acyl group. Now, the last point that I made, in this case, after attaching an amino acid, the amino acid side chain is modified to form the amino acid that is required. And that happens after the tRNA gets attached with an amino acid. The question here is, how do amino-acyl tRNA synthetases attach an amino acid to a tRNA? The process is called as aminoacylation. In the first step, the carboxy group of an amino acid attacks the particular phosphate group of ATP. This results in the expulsion of a pyrophosphate and leads to the formation of 5' aminoacyl adenylate. And this is a high energy compound. pyrophosphate that leaves is also cleaved. So the reaction is driven forward by two phosphoanhydride bond cleavages. The first phosphoanhydride is this, and the second phosphoanhydride is the cleavage of pyrophosphate to two individual phosphates. And the fate of amino acyl adenylate varies. How does it vary? In the first case, amino acyl tRNA synthetases from class 1 use 2' hydroxy group in the tRNA to attack this particular amino acyl AMP, and specifically this carboxyl group that is esterified by a phosphate. Now this particular group is electrophilic, and when the hydroxy attacks this carbonyl carbon, phosphate attached to the AMP leaves. In the second class, the amino acyl tRNA synthetase class 2 uses 3' hydroxy to attack that particular carbonyl carbon of amino acyl AMP. What is the result? The result is the formation of amino acyl tRNA. And in this case, an amino acyl tRNA that is attached to the 2' hydroxy in case of class 1. Now, once this particular compound is formed, it undergoes a transesterification reaction. The transesterification reaction involves this 3' hydroxy attacking this particular carbonyl, because this is an ester, and the 2' hydroxy is then relieved. This results in the formation of amino acyl tRNA, and in this case, the amino acyl AMP is attached to the 3' hydroxy. Alternatively, when class 2 tRNA, amino acyl tRNA synthetase is involved, it uses the 3' hydroxy directly and attacks the carbonyl carbon of the amino acyl AMP and results in the amino acyl tRNA. In both cases, the final product is an amino acyl tRNA. The interaction between an amino acyl tRNA synthetase and a tRNA has been viewed as the second genetic code. Amino acyl tRNA synthetases must be specific for both amino acid and the tRNA. Matching each amino acid with correct tRNA can be viewed as the second genetic code. The code is in molecular recognition of a specific tRNA molecule by a specific synthetase. Only a few nucleotides in tRNA confer the binding specificity. For example, the anticodon region of the tRNA is one specific region that confers specificity. The primary determinants in alanine tRNA is a single GU base pair in the amino acid arm. This was an interesting finding, and we will talk about this in a little bit. Certain nucleotide positions in tRNAs are recognized by amino acyl tRNA synthetases. Some nucleotides are conserved in all tRNAs, such as these that are shown in black, and they cannot be used for discrimination. By observing changes in nucleotides that alter substrate specificity, scientists have identified nucleotide positions necessary for discrimination by the amino acyl tRNA synthetases. These nucleotide positions seem to be concentrated in the amino acid arm and the anticodon arm, including the nucleotides of the anticodon itself. They are also located in other parts of tRNA. Sometimes the T-side arm also has a couple of nucleotides, and D arm has this base pair that is also involved. Now, The positions that are given by the orange and the blue are supposed to be the nucleotides that help discriminate amino acyl tRNA synthetases, various tRNAs. Determination of the crystal structures of amino acyl tRNA synthetase complex with their tRNAs helped researchers in identifying more positions. The tRNA structural elements recognized by the alanine tRNA synthetase are unusually simple. It so happens that a single GU base pair is sufficient for the specificity of alanine tRNA synthetase to recognize the tRNA. A short synthetic RNA mini helix, as shown here, with the critical GU base pair, but lacking most of the remaining tRNA structures, including the anticodon arm, was shown to be selectively amino acylated with alanine. Now, this particular experiment using this short oligonucleotide confirmed the fact that GU base pair is the only structural element in the tRNA that is required for alanine tRNA synthetase to recognize this tRNA. So the fidelity of tRNA synthetases usually involve two-stage proofreading. And in addition to recognizing the tRNA synthetase also has to recognize the correct amino acid. For example, isoleucine tRNA synthetase differentiates between isoleucine and leucine. Let us get back to the 5 stages of protein synthesis. The first stage involves activation of amino acids. And we talked about this in a little detailed manner, how tRNA gets amino acylated by tRNA synthetase. The second stage, initiation of translation, happens. Here, both mRNA and the amino acylated tRNA binds to the ribosome. In the third stage, or the elongation stage, cycles of amino acyl tRNA binding and peptide bond formation happens. Peptide bonds form until a stop codon in the mRNA is reached. And this is when peptide bond formation stops. In stage 4, termination and ribosomes recycling happens. And here the mRNA and the protein that is synthesized dissociate from the ribosome. And the ribosome components are recycled. And finally, the peptide that is formed is folded and sent to various organelles for post-translational processing. Stage 2 of protein biosynthesis involves initiation. And the initiation stage has 3 steps to it. In the first step, the 30S ribosome subunit in prokaryotes bind to initiation factors, IF1 and IF3, at 2 different sites, E for exit site and A for amino acyl site. One of the reasons why these bind here is to prevent this 30S ribosome subunit to bind to a larger 50S ribosome subunit. Following this, mRNA binds to the 30S ribosome subunit, and in this case, the AUG codon is right across the P site, or the peptidyl site. In step 2, IF2, or initiation factor 2, that is bound to GTP, binds the 30S ribosome subunit and recruits a tRNA. And this tRNA is called as the first tRNA, or the FMET tRNA. FMET stands for formylmethionine tRNA. Now, an important point is that in bacteria, the first amino acid of the protein is formylated. And hence the first tRNA always has a formylmethionine attached to it. And this particular formylmethionine with its anticodon interacts with the codon, the first codon AUG, at the P site. In step 3 of stage 2, the initiation factors are released, and this is achieved by hydrolysis of GTP to GDP. And remember, GTP is bound to initiation factor IF2, and once GTP gets hydrolyzed to GDP, the energy released is used to release the initiation factors. Once the initiation factors are released, the 50S ribosome subunit, or the larger ribosome subunit, interacts and associates with the 30S ribosome subunit. And this results in the formation of 70S initiation complex. Now remember, the 50S and 30S doesn't combine to form 80S. As I said before, S stands for sedimentation coefficient and it's not additive. When the mRNA binds the 30S ribosome subunit, the AUG codon is guided to its correct position by a sequence named as Shine-Dalgarno sequence. The consensus sequence is an initiation signal of about 4 to 9 purine nucleotides residues, and this is about 8 nucleotides away from the initiation codon. This sequence in bacteria base pairs with the 3' end of 16S ribosome RNA of the 30S ribosome subunit. And as shown here, this particular region of the 16S ribosome RNA in the 30S ribosome subunit is this. And the blue colored lines are hydrogen bondings. And this is how the Shine-Dalgarno sequence is used to recognize and guide the initiation codon at the right site, and in this case, the P site. Stage 3 involves elongation. And there are 3 steps in the elongation stage as well. In step 1, binding of an incoming amino acyl tRNA takes place. The incoming amino acyl tRNA binds first to an elongation factor named as TU-GTP. A GTP is attached to this elongation factor TU, and this binds the incoming amino acyl tRNA. Now this amino acyl tRNA, or AA2, carries the next amino acid that needs to be added to the first amino acid. So the TU-GTP guides this tRNA to the position that is shown as A, and that's where the AA2 harboring amino acyl tRNA binds. Following this, the TU-GTP dissociates from the ribosome subunit, and the dissociation is propelled by GTP hydrolysis. Once TU-GTP dissociates, another elongation factor, TS, comes in and binds TU, and this results in the release of GDP. TS-TU complex is then further bound by GTP, which binds to TU and forms the TU-GTP complex. So this is a cycle, and this cycle continues in the elongation process, especially the step 1 of the elongation process. Step 2 of elongation involves peptide bond formation. The peptidyl transferase catalyzing this reaction is the 23S rRNA or ribosome RNA ribozyme. The N-formal methynyl group is transferred to the amino group of the second amino acyl tRNA in the A site. That's the A site here. And this forms a dipeptidyl tRNA as shown here at this particular site. Now both tRNA bound to the ribosome shift position in the 50S ribosome subunit as shown here, and this position shift is to take up a hybrid binding state. Now the uncharged tRNA shifts so that the 3' and 5' ends are in the E site. So as you can see, this shift happens. Similarly, the 3' and 5' ends of the peptidyl tRNA shifts to the P site. So this shifts to the P site and each of these shifts to the E site. The anticodon remains in the P and A sites. Now basically what happens is the shift of the position of each of this tRNA happens towards left so that the new tRNA is in the P site with the peptide that is formed. Reiterating what I said before, in step 3 of the elongation stage, translocation of ribosome takes place. And here, the ribosome moves one codon towards the 3' end of the mRNA. So the mRNA codon moves. And in this case, what happens is to move this codon, energy provided by the hydrolysis of GTP bound to the elongation factor G, or translocase, is used. The dipeptidyl tRNA is now entirely on the P site. So as you can see, the translocation, in here it was in the A site, the dipeptidyl tRNA. Now it's in the P site. Now once it's in the P site, the A site is left open. And it is left open for an incoming amino acyl tRNA. So what happens is another tRNA will come and sit in the A site so as to transfer its amino acid to the dipeptide to make a tripeptide. The uncharged tRNA later dissociates from the E site. So this is an uncharged tRNA that is sitting in this site, and this will gets dissociated from the site as a result of GTP hydrolysis. Stage 4 involves termination of polypeptide synthesis. Termination occurs in response to a termination codon or a stop codon. Codons UAA, UAG, or UGA in the A site will trigger the action of termination factors or release factors, RF. First, a release factor, RF, binds to the A site. This leads to the hydrolysis of the ester linkage between the polypeptide and the tRNA. And this ester linkage is between the carboxy terminus and the hydroxy of the 3' region of the tRNA. Now, once this hydrolysis happens, the polypeptide is released from the 70S ribosome. Once the polypeptide is released, finally the mRNA and the deacylated tRNA are also released from the ribosome, which dissociates, and finally the ribosome dissociates into two different subunits, the 30S and the 50S subunits. Now this is aided by ribosome recycling factors. Now IF3 and energy provided by the EFG, that is GTP associated, is essential in this process because The elongation factor G, that's bound to GTP, gets converted to EFG GDP. And the energy released is what triggers the binding of IF3 to the 30S subunit. And once IF3 binds, that releases the 50S subunit. The 30S subunit complex with this initiation factor IF3 is ready to begin another cycle of transition or translation. And if you remember, in the initial step, initiation factors IF1 and IF3 binds to 30S subunit. And following this, when it is ready for the next translation round, IF1 binds to 30S subunit.